As cannabis cultivation continues to normalize across the United States, more and more research and development is being invested in cannabis genetics. Our heritage growers made the cannabis crosses we love today based on intuition and experience. Today, though, a whole new variety of cannabis breeder has evolved, not only using intuition and experience, but also using heavy computing and laboratory analytics as well. While there is certainly no replacement for experience and intuition, technology-assisted breeding greatly decreases the time it takes to actually attain breeding goals because successful cultivars can be analyzed and confirmed on the spot in a spreadsheet instead of having to grow all the plants to maturity and having all the breeder's friends smoke the flowers hoping to confirm the results. With analytical breeding, sex, cannabinoid content, terpene content, and overall potency can be determined in just a few minutes, several thousand times over, making extraordinarily large sifts a real possibility. The application of these technologies does not create genetically modified organisms. They simply allow traditional Mendelian breeding decisions to occur much, much faster. Today we're going to be talking about triploid cannabis genetics. These triploid plants are only available to us due to the availability of computing power to cannabis breeders. These plants have 3 chromosomes instead of 2, which makes them sterile and impervious to unintentional pollination. They also exhibit other traits many cultivators want, like increased yields and resistance to pests and molds. Triploids occur in nature in a great many species, and in nearly all cases, these plants are superior to the other plants in their species, so long as you don't intend to breed with them. If you want to learn about cannabis health, business, and technique efficiently and with good cheer, I encourage you to subscribe to our newsletter. We'll send you new podcast episodes as they come out, delivered right to your inbox, along with commentary on a couple of the most important news items from the week and videos too. Don't rely on social media to let you know when a new episode is published. Sign up for the updates to make sure you don't miss an episode. Also, we're giving away very cool prizes to folks who are signed up to receive the newsletter. There's nothing else you need to do to win except receive that newsletter. This month, regenerative farming nutrient company Everflux is giving away their full line of products to one lucky subscriber to the newsletter. You'll receive a full bottle of their BioFlux fermented plant booster, their bamboo wood vinegar biostimulant, and a big bucket of TerraFlux, their infused biochar blend. You'll get all 3. Make sure to listen to their commercial during the show to learn more. Go to shapingfire.com to sign up for the newsletter this week and be entered into this month's and all future newsletter prize drawings. You are listening to Shaping Fire, and I'm your host, Shango Los. My guest today is Seth Crawford. Seth is a deinstitutionalized academic with a master's in public policy and a PhD in sociology. His focus up till now has been on the political economy of cannabis. For years now, he has been consulted by lawmakers and policy specialists for a better understanding of how the cannabis economy works. Seth is now co-founder of Oregon Cbd Seeds, a company researching, developing, and wholesaling specialty novel cannabinoid-dominant cannabis seeds. Seth appeared on episode 25 of Shaping Fire to talk about the convoluted CBD market, and again on episode 37 to discuss the nexus of the medical cannabis and hemp markets. Today we're going to talk about the dramatic evolution of triploid seeds for cannabis. Welcome back to Shaping Fire, Seth.
Thank you for having me.
Oh, so glad to have you here. So let's get right into it talking about triploids. So, triploids are really common in other agriculture and food industries, but I'm figuring the place to start with today would be if you were to start with the story of your entire valley getting pollinated a few years back, right?
Yeah, that was definitely the impetus for us. This was back in 2016. It was the second year of industrial hemp production in the state of Oregon, and it was the first year of legal adult use outdoor production. So it was the inaugural year for THC, and the first, I would say, big year for industrial hemp. And we had a neighbor who was about 3 miles away from one of our facilities and 6 miles away from our major field, who had about 75 acres of densely planted male and female industrial hemp, or what was supposed to be industrial hemp. he pollinated our field, our facility. He pollinated, neighbors that were 15 miles to the east, 15 miles to the north, 15 miles to the south. It really demonstrated the viability of cannabis pollen and just how far it can travel. So I will grant him that it was a good science experiment. But on the other hand, it devastated a lot of other farmers in the valley. And unfortunately, in ensuing years, it continued to happen in smaller. I wouldn't say it was to that scale, but it's definitely been happening in our valley, and then also throughout the state and other areas, especially in Southern Oregon. It's really demonstrated to people that, even if you have good communication with your neighbors, there are just some folks who cannot be convinced that their right to farm is less important than the common good of everybody else being able to have a crop as well. So it's really an outgrowth of that experience for us.
So folks who are listening to the show who aren't super familiar with pollen, why don't you explain why it's lame that a neighbor's crop pollinated your crop?
If you have an all-female crop that you're growing for cannabinoids, whether it's THC, CBD, CBG, et cetera, or even just for terpenes in cannabinoid-free plants, if those female plants end up being pollinated, you're going to lose about 50% of your overall biomass and about 30% of your total cannabinoid content, just because the female plants are putting all of their energy into that seed production rather than into the stuff that we want. Very, very problematic from an economic standpoint.
Yeah, and essentially you're also ruining good medicine, right? So there's a real human aspect to it as well.
Most definitely. And the other side of this too, and it's going to be a part of this larger story, is, if you're a farmer and you're let's say you're trying to grow a grain crop in the Midwest, and you're trying to feed your cattle a very particular type of grain, in a lot of cases that grain, while the seed itself is highly impacted by the female that ends up producing the seed, it's also impacted from omega-3 and omega-6, and then other components of the seed that is healthy and good for nutrition. That can be influenced by the pollen donor as well. So if you're trying to have a pure type of seed, even if it's just for your cattle, pollen contamination can really play a significant role in undermining that as well. So it's not just cannabinoids; it's also in seed production.
And also, it's even more than just commercial cannabis as well. When I appreciate the spirit of people who want to guerrilla grow cannabis plants, right? Let the cannabis be free. And they make these little fertilizer balls and they chuck them into, vacant yards and, things like that. And the problem with that though, is that if you have these feral cannabis plants growing around, you could have a patient who's got their little home grow with 4 or 6 plants or something. And, they are growing their medicine and it's important. And, somebody has thrown some feral hemp, a few parcels down, and one of those seeds becomes a male, and all of that pollen is blowing windwardly to this patient's house. Suddenly their medicine is going to get all of this pollen and ruin this crop, which could be literally a life or death situation for them. So I'm constantly encouraging people to not guerrilla garden regular sexed seeds, because it's kind of dangerous for everybody.
It really is, and you can't imagine a worse punch in the stomach than to have that small outdoor grow that you put all that time and energy into, in your backyard. In Oregon, it's 4 plants. You get to the end of the season and your plants are finishing up and you realize, oh, no, there's seed in all of this. You basically lose 3 to 4 months of your life and all that time and energy you put into it. It's in a way it's a lot like legalization. At the very beginning, before there was any form of legal THC consumption in the United States at the state level, a lot of advocates would say, we just have to legalize it. Just legalize it. And Well, yes, that's true. We need to legalize it. The process of legalization, once you start to get into the weeds of policies and rules and how that gets implemented, becomes much, much more complicated. I think the same thing is definitely true in industrial hemp. We have a lot of folks that are big advocates of hemp in general who have constantly criticized some of the farmers over the last few years for growing, CBD or other cannabinoids and not putting energy into, fiber crops or grain crops and that type of stuff. And it's that same question. It's, hemp can do these amazing things. However, how can we make all of these different uses coexist peacefully? And I think that's one of the biggest challenges that we have in the 21st century of being able to adapt this crop, both to the landscape and to the social landscape of being able to have people interact in a way that's not going to be negative for either party.
We talked about that quite a bit last time you were here about how, after your valley got pollinated, you tried to pull together the local farmers to create what I called at the time a pollen exclusion zone, for lack of an official term. And you said that, it was actually really challenging because, farmers are some of the most independently minded people that there are, as similarly with, liberation minded folks, and to kind of pull people together to kind of agree to not do certain things on their property. There's a lot of pushback. And, one of the things that we're going to talk about today is why triploids are great. And I think that this is going to be an opportunity today where science is going to make the politics of living in your neighborhood easier.
Exactly. We had tried from a policy perspective and from a social perspective to try to get people to engage and agree and come together, and it wasn't possible. But there's other ways.
So during the introduction to the show, I mentioned vaguely what triploids are, so people knew what they were in for today. But since you are deep into polyploidy science, why don't you go ahead and give us your definition of what a triploid is?
Yeah, and I would first couch this and say the reason that we even have a ploidy breeding program here at Oregon CBD is because we hired a recent PhD, Shen Chen, who is an absolute genius when it comes to both plant breeding and for the manipulation of ploidy. So he's really the one that is the driving force behind this at our company. So Dr. Shen Chen and Brandon Rojas, who works with Dr. Chen, are really the intellectual weight behind this project. So I'm just basically the communicator of general science. So triploids are cannabis is traditionally a diploid species, which means that it has two sets of homologous chromosomes. It inherits one set from one parent and the other set from its other parent, unless it's a self-pollinated plant, in which case it is inheriting from that previous, just from that initial parent. So two copies of chromosomes are great. It gives you, the opportunity, if you have, say, one bad gene or two good genes, to be able to, basically have a fully functional plant, or a plant that ends up expressing things in a really profound way. Triploids are plants that have three sets of homologous chromosomes, which, if two sets are good, three sets ought to be really, really good. But to get to that point, you have to have tetraploid plants. So a tetraploid would be a plant that has four homologous sets of these chromosomes. So what I just described is diploid, triploid, tetraploid. You can continue going up the polyploid scale and having more and more sets of homologous chromosomes. However, as you start to increase those numbers, there tends to be basically a dropping off of the positive impacts and an increase of some of the negative impacts. So one of the challenges in ploidy breeding is to identify at what level of ploidy does this particular species end up having its maximum expression of health, vigor, vitality, etc.
So the fact that these triploid plants were kind of like superhero plants with this extra chromosome really struck me. You know, I understood right off the bat that a triploid was not going to reproduce, right? And therefore, you know, it was not going to be impacted by pollen. And I'm like, you know, cool, Seth, that's a great thing. But then as I researched the scientific papers preparing for the interview today, I'm all like, oh my gosh, the list of increased benefits to plants from triploid, it's really astonishing. So here's a short list of what I've picked up from the science. Pest and disease resistance, stem straightness, increases in chloroplast number, which results in strengthening photosynthesis. Flowers of triploid plants are generally larger and more colorful than those of their diploid counterparts because the energy is devoted to, instead of seed formation, is used for flowers and other organs, height and yield. So, I mean, my God, it sounds like by adding this third set of chromosomes that you're creating these super plants.
It's exactly true. And really what it comes down to, it's a little bit more complicated than that, in the sense that all of these things that you're saying are 100% true. But the challenge for us has been to identify which combinations of genes will result in the attainment of all of those wonderful attributes. What we have seen is that in the same way that you can, take a specific combination and create incredible triploid plants, you can also get the wrong combination of genes and create a plant that is, I would say, less than a diploid in terms of its ability to be very successful in any given environment. So the real challenge this summer has been doing field trials to identify which of those crossing patterns are going to give you the best results.
Wow, that you just put my whole idea on its end, because so technically you could get all of these good variables in the plant, but you also could breed something that's especially bad. It kind of reminds me of, like, maybe with your earlier breeding projects, you were trying to solve a Rubik's Cube, but now it's a cube with 10 rows instead of just 3 rows or something like that.
You're spot on with the metaphor. As you increase the ploidy, you begin to increase the complexity of what you're dealing with. With a plant that is as complicated as cannabis, and up until very recently, so little known about the underlying genomics, it was in a lot of ways a guessing game. And to be perfectly honest, going into this summer, we had ideas about what could work and what should work. And, some of those did definitely have panned out already. But others have been huge surprises. And we basically had to go back to the underlying data that we're able to luckily in-house generate using our whole genome sequence equipment to produce these really high resolution genomes. We can basically figure out, you've got gene A and gene B. We've got 2 copies of gene A and 1 copy of gene B, and you end up with a plant that is 12 inches tall when it finishes. If you get 1 copy of gene A and 2 copies of gene B, the inverse happens, and you end up with a 5-foot tall autoflower plant that's yielding 2.5 pounds of 20% CBDA flowers. So it's really important to be able to get those proper combinations mapped out.
What is the tool that you use to figure out the percentages of variability for all these traits showing up? Because, I'm imagining that triploid kind of makes breeding squares cry, where you're figuring out what traits are going to be passed on, right? I would imagine that triploids makes that very complex, and at some point you want to, just dump all of these data points into a computer and say, okay, figure out of these genetics, which ones are expressing which, how many of them we have are advantageous, and which of these parents we actually want to bring together to secure the most of the traits what we want. Is that simply dropping it into a database, or what is the tool that you use to help move you ahead on that when the variability is so mathematically high?
Two things, and I'm coming from my we've talked about this before, but my PhD is in sociology. And one of the things that we have to do in sociology is take very disparate amounts of data to try to explain a particular social outcome. And in those situations, you end up explaining, you'll come up with a list of variables that are, 8 to 10 really, really explanatory of variables that have a big impact on your final outcome. And they'll end up explaining, if you're lucky, 30 to 40% of the total variation within any given population for any given question. I think the same thing is pretty accurate when it comes to this particular crop. We're dealing with, in a diploid plant, you're dealing with over roughly 30,000 different genes that are in the plant. And you have to deal with, different alleles and different combinations of those in crosses. That obviously is going to increase to about 45,000 when you have a triploid plant. So the tools that we use, the whole genome sequence data is really useful. But at the same time, we're also basically in discovery mode, trying to figure out what each one of those genes are and what they actually do in the plant. Chemical data is also really useful. So we're using genomic data, chemotype data, and then, the last leg of this and triangulating which crosses work the best is just make the crosses and grow them in the field. It's your phenotype data. So for us, I think we can create a broad stroke, broad outline of what could come out of a cross based on the underlying genomics. And we have a really good idea of what the chemical data is going to look like based on those underlying genes. But the actual things that mean the most to us outside of chemicals, the vigor, the flowering time, the ability to deal with plant stresses and, drought, disease pressure, et cetera. Those things really only show up when you're in the field, and it's field trials and growing them out and just visually taking a look at each combination seems to be the best approach. But you really can't do modern breeding without all three. If you're not using all three, you're really just kind of swinging in the dark.
Right on. And it probably also keeps you a better balanced person. Instead of keeping you in the lab, you have to put on your boots and go out into the field.
Exactly. Well, I mean, that's where you want to be in the first place. So it's, it's a good excuse.
One of the things that is of remark in the state of the science right now is, with you trying to activate these different parts of the genome to see what they do. It's as if we're still at a state of science where there's still some intuition involved, whereas a lot of other plant species where triploids are very common and everything's become very mechanized and just kicking out seeds, that science is already settled, whereas the cannabis genome is still so tasty and fresh that you've got your analytical equipment that helps you quantify what's happening. But really, since we're talking about real world expressions that mean a lot in the field, it really takes for you to go out, walk the plants, see the ones that, strike your fancy, touch them, feel them, smell them, and then go back to the lab with that information. And, I like the idea that there's still some analog left in this kind of breeding.
Oh, it's, once you're able to kind of nail down the science side of it and you have a good data collection process going from the genomic side, from the chemical side, and even on the phenotype side, there really is an art to the selection process. There always has been, and I think there always will be. And this is true in any type of breeding program. We're in close contact with a lot of the folks at the USDA ARS here in Corvallis and at the university at Oregon State. Because we're part of this larger plant breeding community at this point, now that there's some acknowledgement that this is a legitimate industry and it is having a big impact on Oregon agriculture and farms across the country. At the end of the day, there's always going to be art involved. You want to take berries, for instance. You want a blackberry that has the right sugar content and the right harvestability and all the right variables. All the boxes get ticked, but at the same time, you also, as a human, you have if you have a fairly developed palate, you can pick you discern between a berry that has just the right amount of sweetness versus a berry that's, say, overly sweet or undersweet or whatever it happens to be. And I think that's where the beauty's at, because everybody has a different interpretation of what that ultimate is going to be, what that perfect version is going to be. And it leads to all of these different spaces being available for different breeders to come in and sort of make their mark.
So before we go to the first commercial break, because I think we're all excited about set 2, where we're actually going to talk about, the science and how they are, triploids are actually made. Since this topic today is actually triploids and not like a profile of your company, is there anybody else in the cannabis industry who is working on triploids that we want to give a nod to as also, being a leader in this area?
Not commercially at this point. However, we are working with the University of Connecticut just as a sort of an informal research relationship. I believe we're a little further along in our process and our breeding program than they are. However, they are definitely involved and doing some really, really cool work. There's it's Jessica Lovell-Brand, I believe, is the professor there. Her and her husband, and then there's 2 or 3 graduate students who are working on projects. They're doing some really cool stuff, and there's also some folks at Oregon State University that are working on this as well. But in terms of commercial adaptation of triploids and being able to release them, I'm confident that we're the only ones right now in the world that will be releasing triploids for 2021.
Man, you're going to be making a lot more commercial seed buyers cry, just like when you brought out your auto-flowering CBD seeds. Oy vey. So
it's the goal every year. You have to get better. If you're not getting better at what you're doing, then there's, I think it's questionable whether or not there's room for you in the space.
Right on. So we will start talking about how these seeds are made after the break. We are going to take that short break now. You are listening to Shaping Fire, and my guest today is socioecologist Seth Crawford. If you listen to Shaping Fire and you grow your own cannabis, chances are high that you are very particular about the inputs you use for growing. People like us painstakingly self-educate on cannabis nutrients and techniques so we can cultivate the best tasting and cleanest flowers possible. And when we go to purchase those nutrients, we want to be sure that our supplier shares our values and is providing exceptional quality. This is why I recommend buildasoil.com to my friends who care about quality. Build a Soil empowers organic growers to do their best work by sourcing and shipping only the finest cannabis growing supplies. From organic inputs, soils, soil testing and pots, to lights, growing tents, sprayers, and cover crops, Build a Soil founder Jeremy Silva doesn't just stock his store with what's available. He goes deep to personally vet each product for quality and determine that there isn't a better version of the product that he could be selling. Because of this arduous process, you know that your options on buildasoil.com have been carefully curated to create the results you are looking for. Not only that, but the Build a Soil way is a philosophy that will permeate your interaction with the company. From website design to pricing and shipping to after-purchase support, Jeremy and his team always strive to do their best and give you the best customer service in the business. Check out buildasoil.com today for top-tier quality cultivation supplies and a friends and family buying experience. And check out their educational videos and extraordinary social media while you're there too. Quality organic growing supplies at buildasoil.com. While I love growing under the sun, there's a lot of good reasons to grow indoors. And if you're like most folks, you want a lighting source that grows high-yielding, healthy plants without using excessive amounts of electricity. BIOS Lighting creates biological lighting solutions that brings the natural brilliance of the outdoors into your grow room. BIOS Lighting has the attributes that I look for in a horticultural lighting solution. I've bought those cheap lights online, and they're difficult to work with and fail in no time. In contrast, my BIOS LED light is industrial grade to last a long time. It is IP66 wet rated, so little foliar overspray won't harm it. It is easy to clean without taking it down, and of course, the most important aspect, it is built for the exact light spectrum I want for great yielding, healthy cannabis plants. And it doesn't hurt that their lighting rigs look badass too. Many horticultural LED lighting systems are based on irrelevant performance metrics, and people love to argue online about these numbers. I prefer to judge on par photon efficiency and how happy my plants are, and the BIOS lights exceed my expectations in these categories. BIOS lights have an optimized broad spectrum that maximizes photosynthesis and plant growth, while also providing the ideal conditions for superior par efficacy and a comfortable visual experience. I also love their attentive and overeducated customer service folks. BIOS starts with a team of biologists before getting the electrical engineers involved. They have studied how light impacts cannabis plants and devised an overall strategy that works. I encourage you to check out their website at bioslighting.com to learn more about how this strategy can work for you. And Shaping Fire listeners can get a special deal. Use the discount code SHAPINGFIRE, all one word, no caps, for 10% off your entire purchase. That's bioslighting.com. One of the reasons why no-till cannabis growing is so valued by farmers is because the mycelium networks in the soil remain established from year to year. And we know these fungal networks are essential because they are the nutrient superhighways that extend far and wide in the substrate to feed your plants. The trouble with growing in new soils or blended cocoa substrates is that it takes most of the plant's life just to create these mycelium highways. Dynamyco endomycorrhizal fungi inoculant reduces that time and gets your plant eating a wider array of nutrients faster. And it's 3 times the concentration of the current leading brand in the US at 900 propagules per gram of 2 fungal species selected specifically for cannabis cultivation. This new product, called Dynamyco, is the result of 30 years of research and trials at the Volcani Agriculture Research Institute in Israel. It has also been vigorously trialed by cannabis and food growers across the U.S. since the product first arrived here last year. You may have already even heard about Dynamyco by its original name, Dynamike. Now, Dynamyco is available at grow shops and online in the United States for the first time. I love using Dynamyco to both speed up the growth of mycelium networks in the soil, but also as a biostimulant to make clone cuttings more virile. You can see side-by-sides showing the comparative growth on their Instagram at Dynamyco. If you demand reliable growing results and appreciate the importance of an active root zone in creating a thriving plant, I encourage you to check out Dynamyco at dynamyco.com and find out where you can get yours. That's DYNOMYCO.com. Whether you are starting with new beds or pots, or if you want to add some zing to tired soil, choose Dynamyco to maximize your plant's potential. Dynamyco endomycorrhizal inoculant. Welcome back. You are listening to Shaping Fire. I am your host, Shango, and our guest this week is socioecologist Seth Crawford. So, Seth, we got everybody all excited about triploids during the first set because we're going to make these, these superhero chemovars that also can't be pollinated accidentally. So, of course, the big question is, how are they made? And I know that there are people listening that are saying, man, F that GMO stuff. And so let's start there by being really clear that this is not genetically modified organisms. So would you go ahead and make a clarification on the science so that people can be really clear that this is actually nature?
These are not genetically modified organisms. It's actually something that does spontaneously happen in nature on its own. And when you maybe step back and take a look at some of the most successful invasive weeds that have propagated across the country, across the world, many of them are tetraploids because of all of those previous evolutionary advantages that we were talking about before in terms of vigor and chloroplast count and efficiency of using resources. So it's something that, in the long span of history happens regularly and has actually given rise to most of the diploid species that we've seen today. So what you end up seeing is a spontaneous transition to, say, a tetraploid, and then sometime in the future, you'll have a halving again and you'll go back to diploids. And it's part of the reason that we see a lot of the diversity in many different plant species is because of these sort of go to tetraploid, go back to diploid over these long periods of time. In our program, we're taking plants that have been very specifically bred for characteristics that we felt were important and are part of our previous diploid breeding program, and now we're bringing in new new characteristics and new traits in diploid form, and then we're putting them through essentially a chemical transformation. So we're taking a diploid plant and doubling its chromosomes. Not inserting anything new, but taking what's there and literally just doubling it using chemicals in vitro, and then having to grow those plants out repeatedly, take cuttings, grow them out again, take cuttings, grow them out again. And at every single one of these stages, it's anywhere between 6 to 8 months to make this happen from start to finish. We have to test every single one of those plants with a flow cytometer, which is a very fancy piece of science equipment that is able to tell you the size of the underlying genome of the plant that you're looking at. So we're having to screen to make sure that these formerly diploid plants have been successfully transitioned to tetraploids.
One of the things I think it's important to point out is that, while you use a lot of technology in this sort of breeding, the use of technology itself does not set it up for being GMO. And the way I normally talk about it with patients is that it's kind of the difference between technology, between a magnifying glass and a scalpel. When it comes to a magnifying glass, you're looking what is there. And then with a scalpel, you are cutting and rearranging. And all of your technology that you're using is essentially different forms of a magnifying glass. So you are using analytics and testing and gene mapping to see what there is so that you can have a better grasp of what's naturally in your plants, so that when you have your plants naturally interact, you get it right the first time-ish, instead of kind of like just shooting from the hip without knowing what's in your genetic makeup like most of us pollen chuckers do. Do you think that's a fair characterization?
Most definitely, yeah. And the magnifying glass versus scalpel is a good metaphor. The way that I usually describe it is we're reading rather than writing. So like our PacBio SeqWell2, it's a whole genome sequencer that allows us to get every single A, T, G, and C that's makes up the entire basis of each one of these individual plants. We're able to read all of those letters, up to 100 times, to know with close to 100% certainty, 99.99% is where the accuracy is at now, what is actually in those plants. And in the case of these tetraploids, you can liken it to a copy machine. So rather than, making some sort of big edits and changing things, we're taking what's already there and just making an additional copy of it. And that's, like I had said before, that's kind of where we get into the nitty gritty, is being able to figure out which of those genes are good, which of those genes, when combined with themselves again, end up being bad, and more importantly, which of the plants in these crosses, which one do you want to have 2 copies of the genome versus which one do you want to have just 1 copy on the triploid crosses?
So this next question is probably going to be dry for some folks, but I know that there are serious breeders listening who are really going to be grateful that we took this extra step down this path. And this is what I'd like you to do, if you would. Since you're since you're saying that each of these cycles in trying to find, tease out the attributes you want take, 6 to 8 months, would you just give us a timeline of milestones on the creation of a triploid chemovar? And what I'm imagining is you're like, okay, we start with this, and then we do this, and then we do this a few times, and then we end up with this. Just because I know that the breeders want to be able to picture the process in their heads so they can wrap their heads around it. So would you go through that process for us?
Yeah. So we release F1s, which means that you have to have two independently developed, highly inbred parents. One is a pollen donor, one is a pollen recipient. Those, just getting to the point where you have developed the two parent lines is, roughly a 3-year process. And I have explained it elsewhere over the years, but it's, you find a cross that you really like. It has the attributes that you want. You inbreed that plant, you take the seeds off, you grow those out, you test them, you find the best one, you grow that out, you self-pollinate over and over and over as many times as you possibly can to lock in all of the traits that are, exactly what you're looking for in terms of stabilization with that one variety. Simultaneously, you're doing exactly the same process with a separate line, inbreeding, et cetera, doing your selections. At the end of all this, 4 to 5 generations down the road, then you can actually bring them back together and create your F1. So you pollinate one highly inbred line with another highly inbred line that results in seeds that would actually go into the farmer's hands and go into their fields that rather than being highly inbred, they're very heterozygous, which gives them hybrid vigor and allows them to thrive in a field. That's a 3-year process. When you add tetraploids to the mix to make a triploid F1, you have to choose, and this is the important part, choose which one of those parents you want to have the 2 copies donated to your triploid. Identify which of those you want, and then go through the process of chemically transforming those plants. And we do that basically from cuttings. And you have to have hundreds of those cuttings, treat them with the chemical. Most of them are going to die. A handful of them will survive, and those survivors end up being the tetraploids that then get cuttings taken off of them, tested again, grown out, cuttings taken off of them again, tested, grown out, and then they become, if they pass all of those tests and remain tetraploids, then those plants will end up going into our production environments to make the triploid seed. So you're really looking at what used to be, if you were really pushing and doing your hybridization programs quickly and still giving yourself a year to do field trials, it's about a 3-year process. Now we're looking at closer to 4.5 years from the time that we start a project until we can get seed into a farmer's hands as a triploid F1.
Well, I like the compactness of that explanation. It's always nice to interview somebody who was formerly a professor. My question for you, though, is so you started this project like 5 years ago?
So the inbreeding to get to the point where we had the diploid parents, we've been working on that this whole time. Basically, where we're at right now is we can take any of these parents that we've developed over the years, and we're continually developing, obviously, new parents. So like this upcoming year, we're releasing CBDV dominant plants and CBGV dominant plants. So new compounds altogether that allow industrial hemp farmers to meet the super stringent USDA enforced 0.3% THC limit, even at harvest. So it's basically a fairly revolutionary development for farmers. So we've got these plants.
Hold on a second. We didn't need to just stop there for a second. All of these growers whose chins just dropped, what you just said is like a bombshell. We've got 2 brand new novel cannabinoids that don't test hot at harvest.
Correct.
That's a pretty big deal to just slide on through. So
it's kind of big. And it's not just like one variety. We've spent the last 3 years working to completely remake our entire library. So all the varieties that we've released over the last couple of years, Suver Haze, Special Sauce, Lifter, et cetera, those have all been converted into these Varin-rich triploid forms. So we're releasing fully compliant fully infertile female flowers. So this is very enriched triploid varieties for 2021. It's very exciting.
All right. So I interrupted you, but essentially you were saying that you can take any of these varieties that you've been bringing in the market for the last few years, do this dance remix on them, and boom, now they can be triploid. And so you're taking stuff that you've already proven and upgrading them essentially.
Yeah, that's exactly right. Exactly right.
So one of the things that I found interesting is that, when you're using these 3 sets of chromosomes, there are all of these attributes, good and bad, that are bouncing around that you're trying to get a handle on. But a lot of these are not good, and they create imbalances, epigenetic changes and genome size and, increase and decrease genomic diversity. What are some of the imbalances that you have seen are common in cannabis to come out when attempting to, find a triploid that's in balance?
The number one thing that we're seeing is, that process of inbreeding. While it does lock in very specific traits that you target, it can also lock in deleterious alleles over time. And what's really problematic is that you don't always see the full expression of the negativity that goes along with those particular genes when there's only 2 copies. But they are coming out when there's 3 copies on a much more regular basis. So you could have a variety that, you've worked over a long period of time to stabilize, and it seems to be, hitting all the marks that you want. But as soon as you add 1 additional copy, then everything goes haywire. Basically, it's that accumulation of negative alleles over time. Now, I could also see this being problematic. We're very specific in flowering time. It's something that we've been spending a lot of time on the genomic side, and just in terms of data collection, being able to catalog every possible permutation that's out there with these different flower time genes and flower response genes that all end up working in concert together to end up forcing a plant to go into flower at particular times. What's really problematic with triploids is that if you get, say, 2 copies of, for example, the gene set that goes along with, say, a haze plant, an old haze that is equatorial in origin. 2 copies of that will, even when you have, say, a copy of an autoflower gene also, you're going to end up with a plant that doesn't finish flowering until December. Right. So it's understanding which combinations end up leading to the results that you want. And for us right now, that is identifying different flowering genes that allow you to have staggered harvests from, say, early August all the way through the end of October, depending on the bioregion that you're in. Really being able to nail that down is critical, but it's also probably one of the most problematic areas, I think, or it will be a problematic area as folks get into triploid breeding, just understanding what those genes are and how those gene networks interact when you increase ploidy.
And when you're using this at a commercial level and you're using thousands of seeds, that becomes really important for the simple reason, if you think about succession planting in your home garden, your food garden, we're planting, lettuces and things that grow quickly, sometimes once a week, maybe every other week. And the idea is that so you get lettuce all through the summer and you don't just get a hell of a lot of lettuce just 1 week, right? And for a lot of commercial folks, they don't want to have to harvest all of their acreage all at the same time. And so they'll plant some, and then a little longer later, they'll plant some more. But that kind of succession planting only really works if those seeds are locked in to go from germination to being finished in a set number of days, say 90 days. So that if you sow seeds a week apart, that they'll be ready to harvest a week apart. And so I can imagine that is probably why you put so much focus on that, because if that doesn't work, then this doesn't work at scale.
That's exactly right. And when you're talking about non-psychoactive cannabis farming, you really are talking about large scale. There are smaller farms, and it's great. We love them. We are doing everything that we can to support them. But on a biomass side, there are really, really large farms. And as a plant breeder, you're standing naked before the world when you've got 1,000 acres of your seeds in a field. And if there's any type of hiccup in terms of that flower response time, you're going to hear about it, and the farmer is not going to be successful. And for us, if farmers aren't successful, then we haven't done our job.
And unsuccessful farmers are nasty. So you want to be real. You want to make sure you do your job well, so that they're not coming with pitchforks and fire torches and such.
Exactly. We'll save that for the government.
Yeah. So let's talk a little bit more about some of the challenges when making triploids. So, albinism is pretty common when making triploids, and that would be no chlorophyll. And so those die pretty quickly because of a lack of photosynthesis. Are you seeing that pop up with working with the cannabis genome? Because it's entirely possible that albinism is more just common in other species.
We do see that, but we have not seen a heightened expression in triploids, and that's the case even on say the poorly performing inbred varieties. It has not popped up. Now, in a field of hundreds of thousands of plants, you're definitely going to see that, and we've seen it in certain crosses in field trials in previous years with diploids. It's not a high frequency, but it's definitely something to be concerned with. The flip side of that, though, I would say, is that if you can lock that trait in, some of those plants that either have albino expressions or have significant extreme variegation, or say even a leaf shape change that is becoming very popular right now, you can make those express in particular ways that would be fantastic for, say, like a home garden. So there's challenges for large scale field production for, say, cannabinoids or fiber or for food, but there's also some really unique opportunities from just a home gardening ornamental plant standpoint that we haven't seen with cannabis before, which is kind of exciting.
Well, here's a non sequitur for you. So if you've had at least one or two albino cannabis plants through all your trials, I would love for you to compare and contrast what you saw as an albino plant versus, very often we will see indoor plants, a photograph posted to Instagram or similar, and there's always the ensuing fight in the thread below arguing whether or not this is an albino plant or whether or not this is a plant that was bleached by being too close to an LED light. And so visually, I'm assuming you've seen these photos too. So,
yeah.
so compare those stark white plants that were grown under LEDs. Is that how a cannabis albino presents itself? Because I know some species look more pink and some look more white. I don't think that I can know scientifically true that I have seen an albino. It's always been someone showing me a picture. So what does an albino cannabis plant actually present as?
What's really interesting is that it very rarely will it actually encompass the entire plant. More often than not, you'll get a sport, a side shoot, or say a half of the really interesting ones are a shoot that half of the plant, or half of the shoot is green and half of the shoot is white. Those are pretty good examples. Partial albinoism. The ones that you're seeing on the internet, if the top of the flower is a half an inch from the LED and the only thing that's white on the plant is that bleached out spot, pretty good indication that it's from the light rather than a genetic abnormality. One of the things that's really cool about especially the variegated plants that have a tendency towards albinoism is 2 years ago we found a beautiful example of this in the field, and Eric in his infinite wisdom dug it up And, I'm like, what are you doing? Why are we spending this time to dig this plant up? So he digs it up, puts it in a pot, and we took it back to our other farm and just stuck it in the shade. He had this idea that, if you just take that plant, get it out of the direct sun and put it in the shade, it would actually start to put chlorophyll back into the leaves. Lo and behold, that's exactly what happened. So essentially what you've got is a plant that thrives in shade-grown conditions rather than a plant that's thriving in these high-intensity, solar radiation in the field, which is really, really interesting. I think it lends itself to the explanation of just how plastic this cannabis plant really is. Give it a different environment, it would thrive, but for field production, it may not necessarily be the best case.
Kind of as a side note, I have been really enjoying seeing all these atypical morphologies of cannabis, kind of have their heyday as there's more people breeding and more people being able to share and feeling free to share their photographs, on Instagram. We're seeing lots more pictures of variegation and the ensuing argument about whether or not it's variegation or whether it's mosaic or something.
Right.
So there's a lot of that, but it's cool to see people sharing pictures of variegated plants that historically we haven't been able to see because everybody is seeing these in hiding, right, during the Prohibition days. And that's really cool. And then of course, like the freak show plant that has got everybody, tantalized for the last couple of years. It looks more like a tomato plant than like a cannabis plant. And then of course the newly arrived Australian bastard, genetic line that again looks like a fern of some sort instead of a cannabis plant. And all these different morphologies, it's, I never thought there was a thing. And now there's these kind of, interesting plants to grow and experiment with for people who just love the cannabis plant itself, at this point, and maybe never, they will never be production level plants. But I really like my freak show that's on my deck right now. It looks super cool,
Yeah. And that's the whole idea. I think, in previous years, if you're capped with a certain number of plants, and you're going to obviously want to cull the individuals that are not going to give you what you need. In most cases that was cannabinoids. Now everything is a little bit laxer, and people are starting to look at the potential of not only the medicine, but also the food and the fiber, and quite honestly the fundamental beauty of a unique individual from a population. There's the entire ornamental plant breeding industry is built on that. And I think we're going to probably start seeing ornamental cannabis plants in people's yards. You may not be able to tell that it's a cannabis plant anymore, but I think that's part of the fun and the beauty and the plasticity of this awesome species.
Yeah, I totally agree with that. That is going to be nice to be able to grow the plant for it, the joy of gardening, right? Instead of feeling like you have to do it in prohibition mindset,
Yeah.
So this next question, I think to a certain degree, I'm asking this next question just because I want to get this phrase into the show. And so there's this thing called Mixoploid Chimera, right? And so a Mixoploid Chimera happens sometimes when breeding Triploids, and its definition is an inconsistent ploidy from one plant, right? And so in my mind, I'm picturing that instead of the breeding process kicking out all Triploid seeds, it's actually kicking out seeds that have got different numbers of genetics in them, different numbers of sets. And so the resulting seed stock is going to be not comprehensively ubiquitous. They won't be the same, right?
Right.
So how hard is it when you are ramping up the scale, right? After you've done this long, arduous process that you've already described to get the plant that you actually want, how challenging is it to then make, tens of thousands or more of these triploid seeds that are all exactly the same so you get the success in the field?
It's a great question, and the answer comes down to just repeated testing. So even though we've, as I described before, you take a significant amount of time to test and test and test over and over using the flow cytometer to establish that you have tetraploids and you have diploids in your breeding population, you have to continue to do that. And it gets even more complicated with what we're having to do for 2021, which is also making these varin-rich plants. In the process, it means that every single pollen donor that goes into one of our greenhouses has to be individually tested, both for its ploidy and for its varin content, to ensure that we've got everything that we're looking for in terms of being able to produce seeds that are, and I wouldn't say 100% uniform on the seeds, because there still is variability. However, they are hitting all the marks for the traits that we're interested in. What's kind of interesting is to actually get to a plant that is 100% uniform would require a different type of ploidy induction. So rather than increasing the ploidy level, you decrease it to a haploid level where there's only one chromosome, one set of, a single chromosome from the parent, from the donor. So that would be an individual haploid that could then be chemically altered to have two copies of those same chromosomes, but they're identical. And that's when you end up being able to produce, they're called doubled haploids, which are essentially seeds that act just like a clone would, but come along with all the benefits of seed vigor and having no diseases, no pests, that sort of thing on the clone itself.
No, that sounds like a gold standard right there.
It really is. And it's, to be honest, I know there are other groups that are working on this. They have been for a while. We have a research contract with Oregon State University working on this just to figure out if it's viable. I think in the long term, it's an interesting area for me, because what you just described and saying, your triploids are uniform. They're uniform to a certain degree. We're very interested in maintaining some diversity within these different genotypes to ensure that if there is some kind of, if you have some sort of environmental problem or you have a pest pressure, you really do need to have a certain level of diversity even in your seed crop that's in a field to be able to handle those types of pressures and to identify, certain genotypes or certain phenotypes that can withstand those types of pressures. So it's kind of that idea of trying to get towards a monoculture crop in the sense of maximizing your productivity of acres, while at the same time balancing that with the necessity to not have a monoculture in your field. Having that enough diversity to be able to respond to short-term pressures.
So let's dig a little bit more into that uniformity question, because, one of the biggest aspects of uniformity in cannabis that's important is with feminized seeds, right? Because it doesn't take many sneaker males to ruin a crop of otherwise female plants. And you're kind of adding another variable that really must be at, if not 100%, really damn near 100%, like we are with feminized seeds, this fact that the seeds won't pollinate, right? So like, how uniform are these triploid seeds that are being produced in their uniformity in they are pollen impervious? Yeah,
it's a really good question. And two questions all bottled up there. One is, what sort of feminization rate are we seeing with these triploids? Is it comparable to the diploids? With our diploids, it's 99.98%. So 1 in 4,000 roughly would end up having a male phenotype. In the triploids, we're seeing exactly the same numbers, which is super interesting to me. And we're going to dig into that a little bit deeper on the genomic side to get a better understanding of it. But the other side of it is, given a triploid plant exposed to pollen, how seed-free is this plant? And what we're saying is that it is nearly seed-free, in the sense that last summer we had an experiment in a sealed greenhouse. This is a 7,000 square foot sealed greenhouse, no air coming in, no air going out, where we had a triploid that was pollinated by, I believe we had 300 different pollen donors that had all been reversed, and they're making copious amounts of pollen. You walk into that greenhouse and you would get sick if you're allergic to pollen. The triploids that were in that greenhouse produced, one produced, I believe, 4 seeds, and another one produced, I believe, 5, which we were comparing it to the diploid sisters right next to it. Those plants were fully seeded, and on a weight comparison, you're looking at about 1/4000th of, wow. Which is pretty phenomenal. The interesting part about that too is that the seeds that come off of those triploids are actually non-viable. So you would end up with, like I said, 4 to 5 seeds per plant in a greenhouse that is completely inundated, like inches of pollen on the benches when we were all done with, at the end of the day. They're highly, highly resistant to pollen, which is really, really interesting, because not only are you protecting your neighbors from, your own crop. If you're planting all female seeds, you're already doing the right thing. But now you're basically guaranteed you're not going to pollinate your neighbors, because the pollen coming off of those triploid plants are also non-viable. There's no risk to yourself. You're not going to self-pollinate because, honestly, the plants don't get pollinated. So it's really a win-win, and they are very, very, very difficult to pollinate.
It's interesting, too, that it's not simply an on or off switch, where the triploids that did produce seeds, it wasn't like, oh, yeah, these produce seeds and now they're full of seeds. It's like, oh, yeah, the plant produced seeds, but it's only like 3 or 4. It means that, even though some of the individual flowers, kind of failed, if you will, to be pollen resistant, it wasn't the plant as a whole. And so there's like multiple layers of threshold there to resist the pollen.
Exactly. Yeah. It's like we'd said earlier, they're super plants.
Yeah, and really interesting to think about. So let's go ahead and take our second break here, and when we come back, we're going to talk about some of the applications of triploids and where this is going to go. You are listening to Shaping Fire. I'm your host, Shango, and our guest this week is socioecologist Seth Crawford. For years, organic cultivators have been looking for a replacement for using peat moss. Peat moss has long been the go-to soil amendment for water retention and container growing, but organic growers know that peat moss is an unsustainable resource, and the mining of peat bogs destroys habitat and releases sequestered carbon. But peat moss works so well that many have continued to use it. But now there's finally a revolutionary replacement for peat moss that shares the same benefits while also being sustainable. Pit moss sounds and acts like peat moss, but instead of being mined from fragile ecosystems, it actually is made from upcycled organic paper and tree bark. Pit moss is excellent at retaining water in your substrate and creating air pockets and tiny living environments for microbes. Pit moss instantly increases aeration, nutrient absorption, and water conservation too. Carefully and locally sourced, pit moss is the result of decades-long research into the use of recycled paper fibers. Pit moss has the fluffy nature of peat moss and handles exactly the same. And like peat moss, pit moss is inert, so it won't change your pH. Available in a range of preparations, including a nutrient-enhanced blend, a cocoa coir blend, and also as an organic soil conditioner with no added nutrients. Pit moss is also available as an animal bedding. So go to pitmoss.com now to learn more. That's PITTMOSS.com. Growing healthier, more sustainable plants. Pit moss. For many, transitioning to organic gardening can be overwhelming. There's so much to learn about soil biology and fermentation. BioFlux Fermented Plant Boost from Everflux simplifies organic farming so you can start growing organically today. Invented by a California farmer growing organic for 40 years, BioFlux is a fermented natural farming preparation for those who want a natural microbe booster without having to brew their own. This extraordinary chemical-free growth and terpene enhancer improves root development, accelerates the conversion of organic matter into humus, increases nutrient use efficiency and uptake, and increases beneficial microbe activity. In addition to the Bioflux fermented plant booster, Everflux also makes an activated biochar called TerraFlux that is infused with the Bioflux plant booster. Imagine combining the buffering and rhizosphere enhancing qualities of biochar infused with a range of earthworm castings, insect frass, kelp and crab meal, oyster shell, and other ingredients. I'm using TerraFlux infused biochar this summer myself, and it smells alive, rich, and potent. These products have been scientifically proven to match yields and increase flower quality and pest resistance when compared to traditional NPK inputs. If you are looking for reliable organic fertilizers that will free you up to focus on other aspects of your garden, consider using the range of all-natural regenerative fertilizers and natural biostimulants from Everflux. Find out more at everfluxtechnologies.com or by following their Instagram at Everflux. For 20 years, Humboldt Seed Company has been family-owned and providing reliable, high-yielding seeds originating in Northern California. While the current trend is to slap one super male into a line of hype strains, Humboldt Seed Company continues to breed with precision and care by doing large sifts and back crosses to emphasize the absolute best traits that a line has to offer. This kind of breeding takes time, talent, and space to work. No matter what kind of aroma you are particularly into, Humboldt Seed will likely have something you'll love. If you love fruit, you can choose from banana, mango, apricot, papaya, blueberry, blood orange, melon, and lemons across their various strains. They have all gas, glue, and classic sour diesel lines as well. Of course, there are the heritage California strains like OG Kush, Jack Hare, and Headband, and their award-winning blueberry muffin is one that delights just about everybody's palate, especially when concentrated. Humboldt Seed Company is proud to bring to market the infamous Freak Show Cultivar 2, which has a great THC high, but looks so much like a fern that some folks can't even identify it as cannabis. It's a plant that really needs to be seen to be believed. If you're looking for well-balanced CBD seeds, Humboldt Seed Company can turn you on to CBD strains that actually have flavor, like the dill and pepper terpenes of Willie G's Lebanese landrace. Whether you are looking for regular, feminized, or autoflowers, Humboldt Seed Company has the gear to make this your best growing cycle ever. Visit HumboldtSeedCompany.com today to check out their line of vigorous genetics, download their catalog, and find out where you can pick them up. You can also check out their Instagram at The Humboldt Seed Company to check out their gorgeous flowers and the extraordinary Freak Show plant. Humboldt Seed Company. Let them know Shango sent you. Welcome back. You are listening to Shaping Fire. I'm your host, Shango Los, and our guest this week is socioecologist Seth Crawford. So, Seth, we've talked about what triploids are and how they're made. But, this conversation brings up all sorts of interesting side questions. And so in this third set, let's hit a bunch of those side questions and start with, it is possible that most people who are not breeding are going to want these seeds as well, right? This idea that they can be, super plants showing all of these incredible triploid, expressions. I mean, me as just a simple patient home grower, I want to grow this stuff, even though I'm not growing acres of it. Are these, is anybody doing triploids that are THC dominant, right? Or is it just other novel cannabinoids at this point, which are being designed for the medical hemp market?
Yeah, it's a great question. And every home grower, once they have grown a triploid, I don't think you will ever grow a diploid again.
That's some fighting words.
I'm not a betting person, but I would definitely wager in that camp. You're looking at a good example. There's really good papers, scientific papers describing the differences between a triploid hop versus a diploid hop. Very similar families. And what you're seeing is essential oils. So the terpenes and the other olfactory components that make up the flavor profile of a hop are more than double in a triploid versus a diploid. The same thing is true in the triploid cannabis plants that we're creating right now. You've never smelled a plant that had a stronger flavor than these triploids. Conversely, they're also producing more cannabinoids. So you end up with a plant it's basically the ultimate expression of what a cannabis plant can be. So to answer your question, in short, no, unfortunately, there are not triploid versions of THC plants that are available right now. But it would not surprise me if there are a bunch of folks who are claiming that they have them after people hear about this, because they are just so awesome. One of the things that we are working on is, obviously we're an industrial hemp seed research and development company, so we have to honor the federal rules and the USDA's guidelines on who we can give seed to and that sort of thing. But I will say that we are pushing very hard at the federal level to be able to offer seeds to home growers. I think we're going to be able to do that in Oregon for 2021, just trying to work it out nationally at this point. For others to participate in that process. But I will say that as part of the research and development process, we end up with a lot of plants that are considered type 1s. Type 1 are high THC plants. In addition, we also have type 2 plants. And this is something to think about from a gene stacking perspective. If you have a type 2 plant that's both has an active CBD and an active THC gene, what does that look like in terms of the underlying ratio when you have 3 potential copies? So you can stack these in different ways to achieve very different results. We're definitely working on making sure that we have tetraploid versions of many very, very famous and useful THC plants. We're allowed to keep those as part of our genetic library, but we're not allowed to flower them out. They would not meet the industrial hemp standards. However, we can do the transformations on site at our company, and then because we live in Oregon, we take these plants home and we can make seed with them and make triploid seed, say in your basement or your garage. We're not doing commercial quantities of that, and we're not legally allowed to sell them at this point, but they're definitely out there, and they are definitely awesome.
So if I understand this correctly, you can develop the genetics legally in your hemp company, but because though that is not a viable product under your license, you have to stop there. So if theoretically the laws were to change and hemp breeding, hemp seed manufacturers were able to now produce type 1s, you actually have got a library of this seed that you'd go, oh, we're just going to put this into production now, even though they're kind of, it's like spin-off technology from the work that you're actually trying to do.
That's exactly right. That's a very nice way to describe it. We are basically just in a holding pattern waiting for federal legalization to open up. We're cannabis breeders, and the underlying chemical compounds are very, very simple targets in terms of breeding. It's the rest of it that interests us. But as good plant breeders, we're always keeping our work backed up and are always ready to move forward when the time is right.
So let's say that you did, take one of these type 1 seeds that's triploid, and you take it home and you make that your patient garden and you grow it and everything's all nice and legal. Are the differences between a triploid and a diploid seed growing? Is it so remarkable that you can tell that assuming that you have some experience growing cannabis plants, right, already, like if I grew this plant, would I be like, holy hell, this is a different kind of plant? Or is it like, it's a better seed? Like how remarkable is it?
In terms of say like olfactory components, not just terpenes. We know now that terpenes make up about 50% of what actually smells in a cannabis plant. The rest of them are different forms of alcohols and especially aldehydes. The triploid plants tend to have anywhere between 30 to 50% increased expression of those particular compounds, which means that they are the loudest flowers that you're going to end up smelling. Just as an example, we've got one of my favorite plants of all time is Chemdog number 4. So it's one of the Chemdog. There's Chemdog D, Chem 91, and Chem 4. Chem 4 has always been my personal favorite in diploid form. Now, when you see it in tetraploid form, it's a whole other animal. normal. Same smell, same giant flowers, same great structure, same over-the-top resin production, but the smell and the trichome content on it, when you look at it under a microscope, or even just a loupe, a jeweler's loupe, unbelievable. Like the concentration is incredible, and the smell is just overwhelming. So you can take that and then say, like in the case of Chem 4, you can cross it with something else that has a completely different flavor profile, say like train wreck, with a terpinolene dominant expression. And then end up creating this new flavor profile that is not only incredibly intense in a plant that's, really, really vigorous, but it's completely unique. It's an expression that has never been seen before because those two types of terpenes don't end up lining up when you're making crosses. So it really opens up the possibility not only of having a more vigorous plant with higher yields and better oil, but also just increasing that total amount of diversity that's present.
Wow, that's pretty sexy. We should have started the show with that question. We would have kept people a lot longer who we lost because of the science. Wow, that's really exciting to be able to try these plants. You must have been flabbergasted because they're like, I know you're you're a breeder and a professor and a business person, but like you're also a toker. You know what I mean?
At the end of the day, I love cannabis.
Yeah, so
really good flavored cannabis.
You must have been blown away to experience Chem 4 as your own triploid.
Yeah, and we're basically doing that process right now. And this is a shout out to CSI Humboldt and Caleb's work. We've been following his work for a number of years and have just always appreciated his level of detail when hunting for different phenotypes and making sure that things are quote unquote real. He does a lot of work making S1s, so taking these really famous plants, these famous cuttings, self-pollinating them, and then offering the seeds. And for a plant breeder, sifting through S1s is where a lot of the magic really happens. You're able to lock in the traits that you really want and get unique flavor profiles. So for a lot of the THC varieties that I personally work with, if it's not a cutting or something that's been shared by other people in the community, we're going to seeds from reputable underground breeders who continue to stay underground, and I appreciate that, who are making these S1s of famous plants. So we've got things like Durban Poison, California Orange, Trainwreck, Purple Urkel, all of these different S1 varieties that have been released, and we're now basically sifting through, figuring out which ones have exactly what we're looking for, both on a genomic standpoint and then from the chemical standpoint. And then we're going to make tetraploids out of them and basically be ready for 2022 when we're hoping that federal legalization happens and everybody in the world can taste these flavors.
Damn, that's exciting. I also love Caleb's S1 packs. Like, people will put out reg seeds and call it a breeder's pack, and, it'll just have more seeds, right? So you get more phenos at a good price. But for me, Caleb's S1 packs are like the gold standard as far as breeders packs. There's so much variety. He describes that he was actually on Shaping Fire episode 49 talking about feminized cannabis, and he's all like, these S1 seeds, if you have enough of them, you kind of have a snapshot of what is possible, in that line of seeds. And it's actually a lot of credit to Caleb as well. You just mentioned him as somebody that you respect, and a couple weeks ago we had Ryan Lee on the show, and he mentioned out of nowhere that he respect Caleb Inspecta from CSI Humboldt as well. So that's that's a lot of good love for a guy who mostly tries to stay in the underground.
Yeah, and I feel bad just for bringing his name up. I'm sorry, but
well, he's not in the shadows. He's just happy to stay small and local to Humboldt.
And it's one of those things where he's actually doing a tremendous service to other breeders by being able to take these famous clones and not just reverse them and make seed, but he's actually doing the due diligence to make sure that it is what it is, and verifying that to the best of his ability, and I think anybody's ability at this point. But then also making something like that available. So, it used to be that we would ship clones around the country in, DVD cases, and that was how the underground stayed alive. It's how we shared cuttings with each other and made sure that there was not only, cannabis plants across the country, and you had sour diesel in, New York and in Oregon. But it was a way of basically creating culture and camaraderie with people who were taking the same type of risks that you were. He's been living that and also making these seeds available from a breeding perspective. If you can get an S1 of, say, Trainwreck, if it's, a legitimate cut, which in this case it is, you have all of the possible permutations of that particular variety in seed form available to you. And because it's an S1, you're actually getting expressions of both of the parents. And you can start to sift through that and figure out, well, what was it that made that particular plant, the original Trainwreck, so amazing? And for us, we're doing it from a chemical and a genomic standpoint. So really exciting. It's exciting to be able to have that as an opportunity, and it's exciting to be alive at this moment in time, being able to subject all of these classic flavors, these classic varieties to really unprecedented analytical capabilities.
This summer, actually, we got a couple of the Skittles S1s from Caleb here on Vashon, and we just opened the packs and we spread them around different patients on the island. And we spread them around before the pandemic got more serious. And the idea that we were going to do was that everybody was going to grow different seeds from the S1 pack, and then we were going to do kind of like a progressive wander your way around the island and check out everybody's gardens and everything. And so we're going to have to do that somehow more creatively with masks and crap this year, now. But, it's really enjoyable, for anybody who loves the plant to check out these S1s, where you're all like, oh yeah, these are all Skittles plants. I can see and smell that, but they all have got, subtly different, personalities. And, if you're a nerd for the plant, that's just a lot of fun.
That I would love to go along on that. It sounds like it's not going to happen this year, but man, what a great idea. Yeah. I love that.
Actually, you're probably going to appreciate it more next year because we also got some of the ChemDog S1s. And so that's awesome. What we're planning on doing next year. So let's talk about something that is really a theoretical, because we'll talk more about, licensing issues here in a little bit. But, I know that the way that your seeds have to be sold, due to the government regulations, is you need to sell to particular farmers and you can't just sell to the public. And, but as we know, occasionally some of those seeds will find their way into a friend of a farmer's hands. What would happen if a home grower tried to replicate these triploids, some pollen chucker?
Oh, is it like pollinating a triploid?
Yeah, totally. Essentially like people were doing with your other autoflower CBD seeds, right? Your autoflower CBD seeds would be put into the market. The farmer who bought them from you isn't supposed to share them, yet somehow miraculously their neighbor gets some, and they hit them with pollen in the basement to try to keep going on the triploid. Yeah,
the unfortunate for the folks that are trying that, you're going to waste your pollen because the flowers are non-viable.
And that's the whole point.
We're not doing it to stop people from breeding with our work. We're trying to help farmers from being, cross-contaminated by a neighbor that's not being wise in their planting decisions, or even, for a farmer that's, say, planting near a feral population of hemp, like we see all over the Midwest, basically from, the southern United States all the way to Minnesota. You're just not going to end up with anything except probably increased allergies if you have viable pollen in your basement.
So what does the future of triploids look like, right? So if we've established that we can do a lot more with 3 sets of chromosomes than we can with 2, And we're just putting our big toe in the pool of triploids in cannabis, and where you're starting is to be pollen resistant, and, you're going for, yield and, pest resistance and drought resistance and such. I'm assuming that there are a lot other traits that can be toyed with, but just aren't going to be in, the first rev of seeds. What do you expect us to see coming from triploids, over the next few years?
It's a really good question. And to be perfectly honest, outside of different flavor combinations that have never been seen, like I was describing before, in these new cannabinoid profiles, your flowering time, disease resistance, those big things that you'd already mentioned, I'm really just scraping the surface. Every single week we're getting another 4 whole genomes sequenced and assembled and, available for us to basically start digging through and mining, trying to figure out which of these different genes are contributing to different aspects of the plant that could potentially be useful. I think in the longer term, the way that I view this, I don't have a specific answer, but I think in the longer term, what we're seeing is that these plants are going to have the capability of having greater evolutionary adaptability. You can go back to the notion of punctuated equilibrium. It's this specific concept of evolution and how it operates. It was put forward by Stephen Jay Gould, one of the most brilliant 20th century evolutionary biologists. Notion that over long historical periods, you end up with essentially stasis, not a lot of change that happens. And then in specific moments, you have what's called punctuated equilibrium. The equilibrium is punctuated by either external events that end up challenging a particular organism, an organism's survival, or something that is localized, say in the soil, or predators coming in and eating the plants. The idea here is that the more adaptability that a particular species has for any of these particular challenges, the more fit it is actually going to be. So as we see, the ravages of climate change or issues with, weeds in fields, or, some of the larger ecological pressures that are undoubtedly going to be coming down on us in the 21st century, these plants have the capability of responding to that in a way that we can't even foresee at this point.
Right on. That's very exciting. And it also sounds like it's a real open-ended kind of answer, right? The answer is, we don't know what might still be in these plants because we're still learning the genome. And also we're not sure what we're going to need them to do yet because as climate change continues, we're not sure how it's going to manifest. So it's kind of like a big open-ended question, which actually makes me feel really enthusiastic for, the future geneticists, and probably even more so the pot smokers that are in college right now who are studying these sciences. What a great time to be a, 18, 19, 21-year-old, something like that in school where you're able to actually study this in the classroom, get into a lab, and then find yourself matriculating out of school and go into legit companies that are working like this and have this be your life's work. I mean, how badass is that? What a great time to be coming into your career in cannabis.
Yeah, and I only got here because I basically got let go from the university for being a proponent of cannabis. So I'm incredibly thankful to be in the position that I'm at. But, the younger folks that we have in our company right now who are recent graduates, with horticulture degrees or botany degrees or plant breeding degrees, they have a really hard time keeping a smile off of their face because this is this is the moment in time. It's this golden age of cannabis breeding and discovery that, you really only get to be around one time, as it's happening, and it's happening. One other thing that may be worthwhile noting is this idea that, one of the problems that the Midwest has, I brought that up. It's good to have a triploid because you can't be pollinated by the feral populations that are left over from, fiber farming in the first part of the 20th century. These native populations have established themselves and basically continue to thrive because it's a fairly adaptable and plastic plant. One of the ways that we can end up dealing with that, I think, over time. And it's a bigger concern here in Oregon, where you had a huge explosion of industrial hemp farms where people were using male and female seeds inappropriately, and then basically just leaving the area. They we're seeing it right now. There's fields all over in the Willamette Valley and in Southern Oregon where the farmers farmed last year, didn't take care of their crop, and they're gone now. But the seeds remain. And so you have male and female populations popping up, wow, creating a feral population where there never was one before. That's a very, very difficult thing to control. One of the things that we're working on is being able to use tetraploids and diploids in rotation. So you basically grow your tetraploids for 4 or 5 years in a specific area, and then you can switch back to a diploid to make sure that you don't have feral populations that pop up. You can use the ploidy to your advantage to basically stop feral populations from emerging.
That reminds me a lot of those mosquitoes that are in the news right now, where they GMO'd up some mosquitoes that don't breed, and they're introducing them to the population. So the mosquitoes are breeding with sterile other mosquitoes. And even though, we're talking about triploids, which are not GMO, we will repeat, the idea is kind of the same. What you're talking about, this this feral reduction, because it'll stop them from producing usable seed.
That's exactly right. And again, just to highlight that, not GMO, we're taking something that naturally occurs. It's just we didn't know it. And to be perfectly honest, I bet there are people who are growing cannabis plants right now in their basement, in their garage, in their closet, that are tetraploid, and they just don't know it, because these plants do pop up naturally in populations. It's just very, very rare, and most people don't have a $40,000 piece of science equipment to be able to test in their home, whether or not it's a tetraploid or a diploid. So it's basically using this new knowledge to really be able to push both farming and ecology forward. We don't have to insert genes from other species to have a good outcome. You don't have to genetically modify organisms to be able to be good ecologists. We can actually use what's naturally available and use the tools that we've learned about in science to make the success rates much higher and much faster to actually push us forward.
Right on. That's beautiful. I don't think I'm going to top that. So as we wrap up here, I want to do something different. You and I have talked, this is your third time on Shaping Fire, and each time you come on, you're usually coming to share some new aspect of science with us. And then after the interview, you get bombarded with emails and contacts and all this kind of stuff, more than you can handle. So why don't we go ahead and answer a couple of the questions that you get hit with? This stuff's all on your frequently asked questions on the website, but let's hit the big two. Like, how do people buy your seed? And I guess that's pretty much the question. And is there any left for this year? Because people are generally pretty disappointed when they find out to get your seed that they generally have to be a prior customer. So why don't you lay that down real quick before we wrap up?
Yeah, it's a great question. I appreciate it. We can always get seed from our website. Not always. We can get seed from our website, OregonCBDSeeds.com. At this point, you have to have an industrial hemp license issued by either your state or by the USDA. There's really nothing that we can do about that. We did stop sales, and we do this every year. Unless you are in a state like California or Florida where there's different growing seasons, we shut our sales down for each year, July 1st, because we don't want to take advantage of farmers. If you're trying to get plants in the ground or seeds started after July 1st in the Northern Hemisphere, you're really missing out. You've missed the boat, and you're not going to have a good crop. So we halt. We'll start sales for 2021 on these new Triploid, Varin-rich, 100% compliant with the USDA 0.3 THC rule on January 1st of 2021. And hopefully by that point, we can have a little bit more clarity from the USDA on who's actually allowed to purchase seed and whether or not we can get these to home growers. So we'll be working diligently to try to make that happen in the meantime.
After you brought your CBD autoflower to the market, which honestly kind of remade the CBD market globally. You sold out of those wicked fast because, of course, everybody wanted tens of thousands of them. Should we expect that with Triploids for the next few years as well? Kind of like we're still seeing with like some of the CBG stuff. Is it, you're here talking about it because Triploids are the cutting edge of cannabis breeding. But is it going to take 3-4 years for enough of them to be produced that they're going to be widely available? How much should we expect on seeing these?
We have enough production capability to take care of the entire country next year.
Oh.
No problem. We've spent the last 2 years focused on building out our facilities and basically taking everything that we made from seed sales and putting it back into science, people, and our production environment to make sure that we could meet those goals and get everybody taken care of.
Right on. Well, congratulations on that scaling, because it was only a few years ago that you were all like, damn, we keep on selling out.
It's true.
And that frustrated people to no end.
Yeah, it's, you do everything that you can to try to meet the demand and keep people happy. But sometimes it's hard. It takes time to build capacity, but we are there, and we're really happy to be able to get people what they need.
Fantastic. Well, Seth, as always, thank you so much for joining us and sharing your very unique perspective from where you're sitting in cannabis. It's always cool to hear about the new things that, cannabis scientists in general are working on. But since genetics is your bread and butter, it's nice to hear what you are going to be bringing out before it comes out. So thank you so much for your kindness and your stories.
Oh, thanks for having me. This is the best show on the internet, and you got some of the best people listening to it. I can't wait for them to hear it.
Awesome. Thank you, brother. I'll see you again soon. If you want to find out more about Oregon Cbd Seeds, you can go ahead and go to their website at OregonCBDSeeds.com. Also, it's great to follow their Instagram at Oregon CBD. You can find more episodes of the Shaping Fire podcast and subscribe to the show at shapingfire.com and wherever you get your podcasts. If you enjoyed the show, we'd really appreciate it if you would leave a positive review of the podcast wherever you download. Your review will help others find the show so they can enjoy it too. On the Shaping Fire website, you can also subscribe to the newsletter for insights into the latest cannabis news, exclusive videos, and giveaways. On the Shaping Fire website, you'll also find transcripts of today's podcast as well. Be sure to follow on Instagram for all original content not found on the podcast. That's at Shaping Fire and at Shango Los on Instagram. Be sure to check out the Shaping Fire YouTube channel for exclusive interviews, farm tours, and cannabis lectures. Does your company want to reach our national audience of cannabis enthusiasts? Email hotspot@shapingfire.com to find out how. Thanks for listening to Shaping Fire. I've been your host, Shango Los.